Welcome to the Virtual Cells Platform AI Workspace demo. The AI Workspace landing page is accessible from the purple button in the top right navigation bar. Sample datasets are preloaded for exploration, or user datasets can be uploaded. Upload options include drag and drop or file selection. Model requirements can be reviewed before proceeding. Create and name a new workflow. Example naming, liver data demo, using a sample dataset. Select a workflow type. Generate embeddings, compared to cell by gene census, or co-embed two datasets. Example, compare to census and set reference tissue to liver. Select a model such as SCVI and run the workflow. Previously completed workflows can be opened to view embeddings. Metadata is displayed, including cell type labels. Color by cell type and add labels. Annotations can be added by creating new categories and applying labels. Autocomplete suggests terms from existing cell types in the cell by gene schema for label assignment. Completed workflows include an Explore button. Explore opens a cell by gene interactive instance. Datasets include categorical labels and continuous metadata such as cell cycle scores. Cell type overlays can be applied to the embedding. Datasets can be labeled directly within the interface. New categories can be created with autocomplete for ontology terms, such as B cells. Labels save automatically and appear after refresh. Categories can be edited, relabeled, or deleted. Existing labels can be refined manually. Labels can be corrected, renamed, and saved to update the workflow. Gene sets can also be saved, for example from differential expression analysis. A UI control agent can perform actions such as coloring by cell type. The agent can select populations, like hematopoietic cells, or create gene sets. Differential expression gene sets can be visualized on the embedding. Genes enriched in specific populations become visible with expected variation. Gene sets remain accessible after refresh. The workspace supports annotation, exploration, and gene set creation for as long as the workspace persists. Deleting the workflow removes the instance and all annotations. Ensure labels are downloaded before deletion as a CSV file.